Thanks so much for having me. My name is Erik Johnson. I'm a research scientist here at the Johns Hopkins University Applied Physics Lab. I work on the BossDB team and our BossDB system, which I'm excited to tell you about today, the Brain Observatory Storage Service. This is going to be more of a talk about computational tools and storage solutions for the EM community, particularly in the neuroscience domain, but potentially has applications across a wide range of different arenas. So, for a little bit of background context, we are part of and funded by the U.S. Brain Initiative with the goal of developing new tools and technologies to understand networks of cells and how brains are organized and behave at a scale never before seen. And a lot of the focus in the first phase of the Brain Initiative was in investing in the tools and infrastructure to really scale up how we go about studying neuroscience. More specifically, we're part of the Brain Informatics program, where a lot of investment has been made in multimodal strategies for understanding the brain. This includes things like light microscopy, omics data, electrophysiology that have been traditionally used for quite a while to study neuroscience systems, but also large-scale electron microscopy techniques with the goal of enabling electron microscopy nanoscale connectomics, so allowing us to really map brain regions at that neuron and synapse level throughout large sections of neural tissue. Within this context, the program has built out data archives, data processing tools, and data standards applicable to these different domains. So with our BossDB team, like I said, we focus on this electron microscopy connectomics domain, and we have built this archive that's specialized within the context of this field to enable large-scale neuroscience studies, but has a lot of lessons potentially applicable to the storage and processing of volume EM samples throughout a lot of different domains. So let me just briefly introduce the notion of a connectomics pipeline. This has a lot of overlap with other electron microscopy volume EM preparations that you're all likely very familiar with, probably more familiar with than I am as a more computationally oriented person. But there are a few wrinkles that are specific to the idea of building EM connectomes. So, like most studies, starts with sample preparation from different regions of the brain, which are imaged in volume slices through a number of different technologies. We try to support a wide range of electron microscopy techniques with our archive. After imaging, these are stitched together and aligned to create a volume. Up to this point, it shares a lot in common with other volume EM applications across a wide range of spaces. Then, from the processing side, we focus on segmentation of objects within the volume, particularly neurons and synapses, as well as a lot of other subcellular structures. These need to be segmented and agglomerated across what are potentially very large volumes. I'll talk about some later that are reaching into the high number of hundreds of terabytes or low number of petabytes in terms of their scale. And after agglomerating these annotations across a volume, find out which ones correspond to cells and synapses, and then use those
Captain,
for connectomics discovery.
what are you
So trying
doing?
to dig into segmenting this into networks of neuron and synapse cells, creating annotations of what things are, and ultimately
Yeah,
wiring
well,
diagrams. That's kind
we
of the
used two different
key
ones. One was
key
just the
outcome
silver,
of these
uh,
neuroscience studies.
like
Where
it,
we're going to get these wiring diagrams through large volumes that show us how all the neurons and synapses are connected within it. This can all get pretty computationally intense. There's a lot of steps to this process, both manual tools to do this at scale, as well as automated tools that incorporate AI and machine learning techniques to automate different steps of this process, are all really critical as we try to scale up to millimeter or larger scales of volumes that could include hundreds of thousands of neurons and millions of synapses. And so a lot of the talk here is going to focus on the data ecosystem that can enable all of this analysis after the image volume has been created for these neuroscience samples. And that's really where our BossDB team comes in. So a bit of an overview of BossDB and who we are and what we do. BossDB is a volumetric image store. We store a wide range of data types, focusing on high resolution and large scale data sets, which makes it ideal for large volume EM samples. It's optimized for efficient cloud storage and sharing of complex scientific data. And it has processing and analysis tools that are really focused on this high resolution EM domain. And so this is a cloud-based infrastructure that's freely available to the community, and is a great resource for researchers that need a robust infrastructure for large data set storage, archiving, and sharing. And it is aimed at enhancing our collaborative efforts and analysis across a wide range of data sets through community efforts, emphasizing easy access and data management for releases and versions of your data sets. So a few things. You can go right now if you're interested to bossdb.org and kind of follow along as we go through this talk here. But a few of the things that I'll highlight here. We have projects that incorporate a wide range of different volume electron microscopy techniques, as well as X-ray microtomography and nanoholotomography approaches. If you go to our projects page, you can get a sense of the wide range of things that are supported. Each page has a lot of detail on the individual efforts, data sets, and analysis to kind of give you a quick overview of all the different things that you can find within the system. So this is a cloud-based ecosystem. We're mostly using AWS as our backend. It stores volumetric data, so not only the raw images, but like I talked about, all of the other things that are generated as part of this data ecosystem. So that's the segmentations of the neurons, annotations of objects, 3D meshes of those structures throughout the volume, and the actual connectomes, the graphs, these neuron synapse wiring diagrams that are created. The archives are just a little north of 1 petabyte of compressed data currently stored across these projects. That's 13.5 petavoxels. So we're getting pretty big. We have a range of model organisms, both invertebrate and vertebrate, and a couple hundred user accounts. In general, all these data sets are stored and publicly accessible. So you don't need a user account to access the data, visualize the data, and get started checking out the BossDB system. These data are divided over 55 public projects that include a bunch of different individual experiments and channels, which I'll talk about a bit more. But the projects work to kind of aggregate that together into cohesive things, often based around papers that have been released and published. You can go there. There's search functionality for species, imaging modality, locations, and keywords, kind of all your usual stuff. If there's things or systems that you're particularly interested in, like I said, focused on the neuroscience domain, but if that's your area of interest, please check it out and see if there are data sets you're interested in. Most of these things are licensed under the CC BY 4.0 Creative Commons license, which makes it open source and available with appropriate attribution. So you're supposed to cite, the data set and the researchers and papers that generated it. And we also mint DOIs so that you get a data set identifier that's permanent and associated with your data set for all of your citations, sharing, publication, and other types of documentation needs. So this is all enabled by, like I mentioned, our cloud-based ecosystem. So over on the right, you can see, kind of a little rough breakdown of the system. If you're a computer scientist, maybe this kind of thing is interesting to you. But just as a general idea, we use Amazon backends for data storage and managing access. There's an API that controls users accessing the data. So in general, you don't just need to download a bunch of unstructured files, but we provide a bunch of functionality so that you can interact with it in different ways. From the perspective of the end user, the key things that you're able to do, you can ingest your data. So if you have volumes that are ready to go, you can get them uploaded into the BossDB system and share them with the world. You can visualize data that's in BossDB in your web browser, as well as pull down individual images as well as volumes on demand that you're looking for. So if you're looking for particular images or 3D volumes, subvolumes from a larger experiment, which is what you're interested in analyzing, we have tools to kind of pull that all out quickly. In addition to that, we're building out a variety of tools for analysis. I'll highlight a few of them that are kind of ongoing. As well as we have a metadata service and data standards and data model that I'll talk about a bit that we enforce in the archive to help make sure that things are standardized and ready to share with the goal of enabling secondary analysis and dissemination throughout the community so that your research not only benefits you and gets you your papers, but can also have impact for years to come in the broader community. As I mentioned, we support a wide range of different data types and species. And I'll highlight a few different tools that we have to help access and manipulate these kinds of data. The core functionality is uploading subvolumes and secondary structures to the data set and then pulling them into different tools for visualization. NeuroGlancer is our most commonly used tool for visualization, but we also support virtual reality 3D visualizations, as well as a lot of tools like Newprint and Motif Studio for looking through the annotations and generated graphs for secondary data analysis. If we look at how projects are actually structured within the BossDB system, we have a notion of a project that bundles together a set of data and metadata, usually in support of a paper, although some of our projects support mini papers because these volumes tend to be very rich and used for many different contexts. Within a project, we have a notion of collections that group together a set of different experiments. Each experiment is associated with a three-dimensional coordinate frame. So typically an experiment will be associated with an individual volume. A collection will bring together a set of related volumes, and a project may have multiple collections. So for instance, if you're gathering volumes from different species to compare against each other, each volume would be a different experiment, and then you would kind of lump those all together into a collection for comparison. Coordinate frames establish the bounds of a volume, so what its size is and resolution, all that kind of important stuff. And then individual channels store the different types of data that we need. So obviously there's the raw imagery itself, in addition to all of the annotations and secondary data products that are available. Critically, these channels also support different bit depths as well as resolutions in a very dynamic way, which lets us do visualizations efficiently across different scales. So you can visualize at low resolution, very large regions, and then zoom in very dynamically on smaller regions that you need. So if we look at a example project, this one is high resolution 2D TEM images from the amygdala of mice, organized as a single collection to support a publication. Um, this includes multiple experiments, each with its own coordinate frame because it's coming from a different part of the brain. And then hundreds of individual 2D images are stored as channels and have their own coordinate frames and associated annotations. And so this was a project focused on synaptic analysis in Parkinson's disease. And so this is kind of an example of the type of project that many people in the community create that BossDB can help support and share with the world. And so you get your project page, you get your DOI, you have all your data hosted. There's example code and visualizations to help people get started with accessing and visualizing your data set. And this is something that's publicly available that you can share with collaborators, reviewers, program officers, all those sorts of folks. And so this comes together where these different samples, like I mentioned, are associated with different coordinate frames and then different channels. And then within each channel, you can have not only the electron microscopy data, but the different associated annotations. Now, this can scale up to quite large and ambitious projects. So one that some folks may have been aware of from a recent Nature publication package that came out a few months ago is the Microns project. This is a full cubic millimeter volume of mouse visual cortex with a resolution of 8 by 8 by 40 nanometers, containing about 75,000 neurons and a whole bunch of synapses, about 1 petabyte of data in total. So this is organized as a single collection and a single experiment with a single coordinate frame because it's one massive volume and includes the image data as well as the annotations for the segmentation of the neurons, the synapses, and the nuclei that have been identified throughout this volume. And then a live complementary tool of all of the ongoing annotations that are being iteratively proofread and improved over time is also available for this data set. So this is a very large and complex data set that can be hosted and visualized through BossDB. And so this is pulled together, like I mentioned, into a single experiment with all of these different sub-channels to organize the different subsets of data. And so large single volumes are also very well suited for BossDB. We have a metadata service. So this standardizes all of the information related to a project. So all of the stuff related to your experiments and preparation, as well as the information about the authors and study participants, funding information, all that kind of stuff. This is available. It's also searchable, and there's an API for querying, so it can hook into other tools and portals so that people can programmatically access and display information about what's available on BossDB. In terms of data access, we support a number of different ways of accessing the data, but one of our common ways to do that is our intern API. So this is a Python-based way to download data and upload data. And we provide code snippets for all the data sets that are uploaded. We have a variety of core services that support uploading your data, managing your metadata, pulling down individual tiles and volumes, pulling down objects, and doing all this visualization. And this is what we use to build all of our tools on top of. And we have a variety of tools that we support in BossDB. So in the upper right here, you can see NeuroGlancer being used to visualize different 2D slices as well as 3D meshes throughout a volume. This runs in a web browser and links are generated for all the projects that are uploaded. We've also done different types of 3D visualization. So on the left, you can see integration with the SciGlass system. for seeing 3D volumes in a virtual reality environment, as well as an ingest of three-dimensional meshes from a worm C. elegans data set. Um, so these allow you to do a lot of different types of data visualization. We also have tools for data annotation. I'll go through one called Bossy Paints, as well as ongoing work into automated segmentation workflows. So being able to automatically segment out things of interest within the data set, such as synapses and neurons. If we look at our annotation tool, Bossy Paints here, this is a web-based tool that allows you to put in a URL to a data set you care about. You can get those off of our website. They're already generated. You can select the volume that you're interested in, and it loads it into your web browser. So you don't have to install anything on your computer. You can take a look at the volume. You can find something of interest like this mitochondria and label it throughout the volume, going through each slice and coloring it in with our little tool here and a little autocomplete thing that lets you label the volume as you progress through it until the object disappears. And then you can scroll back and forth, make sure you find your object of interest in segmentation. And then you can save that out and go on to your next objects. In addition, you can put together a queue of tasks and share this out with different people. For instance, if you have multiple folks within your lab working on annotations, you can have lots of people accessing this data set with a queue of tasks that they need to be working on. We've been working on a few additional tools. One that we are working on is an AI assistant tool. So this is a customized chatbot focused on BossDB datasets that can be used to find out more information about datasets within BossDB. And then you can ask it, native natural language questions and get responses of different types of code and tools that you can use to access and manipulate your data set. So this is another service that we're working on that is now live, that is something we're adding features to that we hope will be useful to helping people get started, especially folks with less of a background in the coding domain. We have a lot of tools for secondary data analysis as well. So once the data have been ingested, visualized, and annotated, there's a lot of different types of analysis that are done in the connectomics community. That can include going through our graphs, our neuron synapse graphs, and learning about their properties. We have a tool for that called Motif Studio. We also have worked with Howard Hughes Medical Institute Genelia Research Farm tool called Newprint that allows you to do queries on annotations generated throughout a data set, and other tools that can be used for analysis of neuron morphology and connectivity. BossDB has a wide range of users. We have a number of dedicated user accounts, and like I mentioned, we also make the data publicly available. You can access all the data without an account. So we've grown a lot in use and activity over the years, and we have not quite a petabyte of data download, but more and more folks are finding data sets are useful for secondary analysis, and we're starting to sort of disseminate this not only around the United States, but around the world. So if you're interested in getting started in BossDB, I encourage you to go to our website. Like I said, you can go to our projects pages. That's a great place to get a sense for the type of data and tools that we can offer to potentially support your projects. If you're interested in uploading and sharing data, or you're interested in different tools that could be applied to your data, you can go check that out. If it seems interesting, you can dive in and create an account and start working on creating projects. We focus on neuroscience data sets. That's our strength. So any 2D or volume EM samples of neural tissue, or different types of associated tissue, neuromuscular junctions, those kinds of things would all be a slam dunk. But we're always interested in talking to people doing more general volume EM studies, to see if there's things and tools that we can offer that can meaningfully support your work. So with that, I'd like to wrap up. Thank everyone on our team, the amazing people that build the archives, the tools, and interface with data collectors to help get their data into BossDB. Our funding, of course, from the Brain Initiative and the Brain Informatics program that's been instrumental in helping stand up this archive, and our range of collaborators throughout academia and the corporate world that have really helped build out this archive and everything that it can do. So with that, I'm happy to take some questions, and I'll be lurking in the chat for at least 10, 15 minutes after here.